Medical students, welcome to today's lecture on molecular biology. We will be diving deep into the intricate world of cell signaling pathways. Specifically, we will explore 12 different signaling pathways that play crucial roles in various cellular processes. These pathways include G-protein coupled receptor, GPCR, signaling, receptor tyrosine kinases, RTKs, second messenger systems, protein kinase cascades, crosstalk and integration of signaling pathways, G-protein coupled receptor kinases, GRUKs, and arrestins, GT-PASase and small G proteins, phosphoinositide 3 kinase, PIEK, signaling, nuclear factor kappa B, NF kappa B, signaling, Notch signaling pathway, WNT signaling pathway, and hedgehog signaling pathway. GPCRs are a large family of membrane-bound receptors that are involved in signal transduction. When a ligand binds to the GPCR, it triggers a conformational change, allowing the GPCR to activate G proteins. These G proteins consist of alpha, beta, and gamma subunits. Once activated, G proteins regulate various intracellular signaling pathways, leading to physiological responses. RTKs are transmembrane receptors that phosphorylate tyrosine residues in response to ligand binding. This phosphorylation event leads to the activation of downstream signaling cascades, including the Ras/mitogen activated protein kinase, MAPK, pathway and phosphoinositide 3 kinase, PIEK/ACT pathway. RTK signaling is crucial for cell growth, differentiation, and survival. Second messenger systems are essential for signal amplification and transmission. Common second messengers include cyclic adenosine monophosphate, CAMP, cyclic guanosine monophosphate, CGMP, inositol triphosphate, IP3, and diacylglycerol, DAG. These second messengers regulate the activity of various protein kinases, ion channels, and transcription factors, thereby modulating cellular responses. Protein kinase cascades are signaling pathways that involve a series of phosphorylation events. These cascades often culminate in the activation of transcription factors, which regulate gene expression. Examples of protein kinase cascades include the MAP pathway, C-Jun N-terminal kinase, JNK pathway, and P38 MAP pathway. Cells integrate multiple signaling pathways to generate appropriate and coordinated responses. Crosstalk occurs when signals from one pathway influence the activity of another pathway. This integration and crosstalk play critical roles in the regulation of cell fate decisions and the balance between cell survival and apoptosis. GRUKs phosphorylate activated GPCRs, leading to their desensitization and internalization. Arrestins then bind to the phosphorylated GPCRs, preventing further G protein activation. GRUKs and arrestins play crucial roles in the termination of GPCR signaling and the regulation of receptor responsiveness. GT-PASase, such as RAS and RO, act as molecular switches to control various cellular processes. They cycle between active, GTP-bound, and inactive, GDP-bound, forms, modulating the activity of downstream effectors. Small G proteins, including RAS and RO family members, are involved in signal transduction, cytoskeletal dynamics, and vesicular trafficking. Peak signaling is crucial for cell growth, division, and survival. Activated RTKs and GPCRs can activate peak, leading to the generation of phosphatidylinositol, 3,4,5 triphosphate, PIP3. PIP3 recruits and activates protein kinase B, PKB/ACT, which regulates various downstream targets involved in cell metabolism, proliferation, and survival. NF kappa B is a transcription factor that regulates genes involved in inflammation, immune responses, and cell survival. NF kappa B signaling can be activated by various stimuli, including pro-inflammatory cytokines, pathogens, and cellular stresses. The activation of NF kappa B involves the phosphorylation and degradation of inhibitory proteins, leading to the release and translocation of NF kappa B into the nucleus. The Notch pathway plays crucial roles in cell fate determination and tissue development. Notch receptors interact with ligands, leading to proteolytic cleavage and the release of the Notch intracellular domain, NICD. NIC translocates to the nucleus and interacts with transcription factors, regulating the expression of target genes involved in differentiation and cell proliferation. The WNT pathway controls diverse cellular processes, including embryogenesis, tissue homeostasis, and stem cell maintenance. WNT ligands bind to frizzled receptors and co-receptors, leading to the stabilization of beta-catenin. Stabilized beta-catenin translocates to the nucleus, where it interacts with transcription factors to activate WNT target genes. The hedgehog pathway plays critical roles in embryonic development, tissue repair, and cell differentiation. Hedgehog ligands bind to patched receptors, relieving their inhibition on smoothen. Activated smoothen then initiates downstream signaling events, resulting in the activation of glioma-associated, Li, transcription factors and the regulation of target genes. The understanding of cellular signaling pathways is crucial for unraveling the complexities of physiological processes and the development of targeted therapies for various diseases. By comprehending the intricacies of GPCR signaling, RTKs, second messenger systems, protein kinase cascades, and other signaling pathways, we can gain valuable insights into the mechanisms underlying normal cellular function and pathological conditions. Stay curious and inquisitive as you embark on your medical journey, continually seeking knowledge in the fascinating field of molecular biology.